for whole genome analysis. Additionally, an increasing number of researchers wanted to analyze their data themselves, and they didn't have enough computational capacity to do it. With the VSC, we were able to move some of our complex research projects to the cluster at the VSC, alleviating the internal load of our own systems. We have successfully performed de novo assemblies, long-read iso-seq analysis, and RNA-seq analysis. We have also guided our research users on the use of the VSC to perform complex genomics analysis, which enable them to advance their science. The analysis of sequencing data is a demanding computational task. The VSC offers all computational capabilities to analyze such a data efficiently and with excellent assistance from the support team. If your research is in need of such analysis, the VSC would be my first recommendation.
My name is Vries and I'm a climate scientist working at the Vrije Universiteit Brussels. In our team at VUB, we use global climate models to study how climate extremes will evolve towards the future, and also to better understand the role that the land plays in shaping these climate extremes. So in our group at VUB, we use the Community Earth System model, which is a global climate model that represents all these climatic processes that occur in the atmosphere, the oceans, the land, and the ice sheets. Now, as a group, we contribute to improving the quality of that climate model by introducing and improving the representation of water management and land management in that climate model. For instance, we incorporate reservoir management or irrigation techniques in that climate model. And by doing so, we improve the quality of the model, we make it more realistic, and this helps to generate more reliable future projections of climate change. So to run our climate model, we need access to high-performance computing infrastructure. And when we have that access, we can contribute climate simulations to the international climate science community. We can thereby support the development of the IPCC climate reports and also contribute to efforts such as the one shown behind me, where we quantify how many climate extremes a person born in a particular year in a particular country will face integrated across their lifetime. So doing this type of supercomputing simulations allows us to make international contributions to climate science. VSC is critical for performing our work as global climate modelers. With the help of VSC, we can run our global climate model on the supercomputer, and we can store the output on a shared data storage facility for further analysis. We are also collaborating with a whole range of other research groups in Flanders to share these resources for climate science analysis. And by doing so, we can improve our understanding of climate change and we can help contributing towards solutions.
Today we are here at the Faculty of Veterinary Medicine of Ghent University. My name is Sebastiaan Deuns. I'm a veterinarian and co-founder of PathosSense, which is a new spin-off of Ghent University. With PathosSense, we offer complete diagnostics of infectious diseases for veterinary medicine. In this, we use nanopore sequencing, a revolutionizing way to analyze DNA and RNA of different kinds of organisms, including viruses and bacteria. Now today, Nick Verreke, my colleague, will take you along in his research in which he is combining nanopore sequencing and a lot of different computational analysis for which he uses the Flemish supercomputer.
We are working with nanopore sequencing. We started with this small MinION device, and recently we moved to the GridION, which allows us to generate a lot of more data. This sequencing data can be used for bacterial identification and antimicrobial resistance testing, which later on can be used for quick and real-time bacterial diagnostics in veterinary medicine. Could we have done this without the use of the HPC? Probably yes. While we started with a CPU-driven computer, we quickly went to a GPU-driven one. However, this did not sufficiently speed up our processes. So we went to the HPC, which could actually facilitate our programs and algorithms to speed up our analysis even further. VSC allows us to focus on the science and less on the computations, so we can improve our portfolio and the number of analyses within the spin-off company.
Thanks to the Flemish Supercomputer Center, we have been able to bring our research to the next level.
Perfect. Okay. So let's start. So thank you very much for this invitation. And what I will like to do today is maybe introduce how AlphaFold might impact structural biology, but generally biology, and what are the challenges, and how we might think about addressing some of those challenges. So I want to start from 1970s because I think the foresight that structural biology community had to create a database and put all their data freely available in this database is really key to all the developments that have happened. So PDB was established in 1971 with 7 structures. Today we have about 200,000. It's now a global mission to actually maintain this with the WWPDB consortium, which has partners all over the world. And I think we need to celebrate this as a starting point of trying to understand biology at molecular level, at residue level, understanding function. And the impact of that started very, very early on. So it's not that we had to wait for a long, long time to get to that impact. So the first, I think I just want to highlight is the seminal paper from Chutia and Lesk around 1986, where they realized with very few structures in the PDB that the structures are a lot more conserved than sequences. And that's the first, I think, insight into how the relationship between sequence and structure works. And then that led on to, obviously, homology modeling. But I want to highlight the first kind of homology model publication that I found, which was even before PDB was established. So this is from 1969 when this model was built by hand. And obviously the availability of PDB made it possible to develop many softwares in the 1990s to do homology modeling and get structures where the sequences are very close to something that is in the PDB. And then I think one of the other insights that's really critical for this whole story is in 1994 paper from Alfonso Valencia and others who were at EMBL at that time, that if we look at sequences, and the hypothesis was that as sequences evolve, there must be constraints on certain residues to co-evolve, and because they might be in contact in 3D structures. So obviously, if one of those residues changes, the other one has to change to compensate for the change in the first residue. And this, I think, insight was extremely important. Going from here, you see a lot of methods developed based on this insight. And obviously, AlphaFold and RosettaFold and everything else also uses this to seed basically the structure prediction process. So there are two elements that were critical in getting this recent advance on AlphaFold and RosettaFold. One, open data and the diversity of data that was deposited to PDB. And the second is the sequencing. So both these together have helped in getting AlphaFold and RosettaFold to where they are today. But I think it's also important to recognize that in isolation, many different teams are working and developing methods, but you need a framework where they all can come together and they can exchange knowledge. It's a competition, but it also becomes a collaboration because you discuss things and then you go away and you develop your methods. And so it's just the way this whole community works together through the CASP that John Moult established in 1994 is important to recognize as well. And there are many similar efforts and frameworks exist today, including Capri. So Shoshana is here today, who now spearheads Capri, and there are other such efforts as well. And this is where you see a community built and knowledge exchange happens, and obviously everybody benefits from such exchange. And then that's what led to 2020 CASP, where AlphaFold was basically above everybody else in terms of its prediction ability. And then 2021, as was mentioned, this paper came out. So the reason I wanted to put this here is for two reasons. One, I think, is obviously this is the first time AI had shown that it could solve a problem that has been around for a long time. And the second, I think, and even more important in my opinion, is the details that were made available when this paper was published. So I forget all this. I think the most important thing on this slide is that supplementary material. And this is what helped all the efforts in OpenFold and everything else, because every possible detail was made available. And that means now many more researchers can look into this and really try and understand what is important, why is it working, how it is working, etc. So let's go through some of these different aspects of AlphaFold, the whole pipeline. The first is inputs, and obviously the multiple sequence alignment is the most important input to this method. There isn't any tricky things here. It's all standard software used to just collect sequences and make a multiple sequence alignment. As a rule of thumb, you need to have enough sequences to seed this coevolution information, which means about 30 or possibly more than 100. The input sequence is obviously the other part that goes in, and this is what you are then creating this matrix. And what throughout this process, what you want to do is basically fill in this matrix to say what is the relationship of a residue pair. And that's what this whole pipeline is about. And the third aspect of it is you can give a template. And obviously, if you give a template, what one might think is that does have impact, but it's not very clear how it impacts or whether it impacts at all. Obviously, there are papers which now show that if you don't have MSA, then template does have impact, and AlphaFold does take into account that particular template. And so it's really important to understand why we are giving certain inputs and what impact they might have on the prediction that we get at the end of it. The second bit of it, which is the kind of black box, in the sense, I haven't seen exact explanation as to how this works and why it works rather than how it works. But what you are getting here is you are filling in this pair representation, and you start from the MSA, you have certain seed, but as you go through the pipeline, you are filling in more and more details, and you're going beyond just the couple of coevolution pairs you might find, and getting the relationship between these different residues, which obviously then you can put into the structure model. And what it does is basically all the residues are at the origin at that point, and now it knows what the relationships are. It transforms them and creates a structure, and that's your starting prediction. And there is another network that also does the side chain, and so then the final structure is achieved through some relaxation, because obviously just rotation and translation of residues from origin to their place is not going to give you a proper protein structure. You need to have proper bond length angles, etc., where possible. And this is what this relaxation process is doing, but it's heavily constrained by the relationships that are found in this matrix. And so it's not a normal relaxation process. It's a heavily constrained just to make sure that the final structure resembles a protein structure. And then obviously what you can do is you can have these evoformer blocks and you can have many of them, or you could take this initial prediction, you feed that back in, and that means you are not just repeating these evoformer blocks, you are providing it with additional information. So the starting point now is a updated matrix and the MSA, and which means the next block is going to have a much better starting point than if you just had these blocks one after the other. And so this is my thinking that the recycling works, because every time you start recycling, you're starting from a much better place in terms of getting the final prediction. But all that, if we think about it, maybe homology models were as good. They had their own limitations, but the models were good, right? So if you had a 3DB model, then you could obviously get a homology model that was good. So what is so special that makes these new methods so useful? And I think the most important thing that comes out of it are these confidence metric. And that's what makes these methods really useful, because we are not only getting the predicted model, we are getting what is the confidence of the particular network in those models. And so we'll go through some of this. And I think that's in my opinion, that is the revolution. So it's not just the prediction of model that is the revolution. I think the revolution is you are predicting model, but also providing confidence of that particular network in those models, which means we start with a much better understanding of how to use these models as well. So let's go through the two metrics that it produces. One is the predicted local distance different test. So LDDT has been around and has been used for assessing models even before AlphaFold. And what AlphaFold does is it predicts what would be the LDDT. And this is a local measure. So you are basically trying to understand, is the packing good? Is the local environment of the residue good? And that's what this is about. So how can you use it? So AlphaFold gives PLDDT in a range of 0 to 100. So anything that is above 90 or above 70 is a good predictor. So if it is above 90, even the side chains would be in really good place. If it is above 70, you can rely on the backbone, but you have to be careful about the side chain. And then you have PLDDTs below 50, which analysis has shown that they resemble the part of the sequence which is intrinsically disordered. And so one of the questions is AlphaFold predicting intrinsic disorder? And the answer is, at least in my opinion, it's not. What it is doing is it is so good at predicting the structured part that as a consequence we get a region which is not structured, and some of that might be intrinsically disordered regions. So not all of it will be intrinsically disordered regions as we know it. So this is the way you would use PLDDT. And then the question is that enough? And I have seen many people in many papers only use PLDDT. And I just want to go through the PAE and why it is so important that you use both the PLDDT and PAE. So the problem is we now have confidence in individual residues, but we also need to understand what is the confidence in a pair of residues. What is the relative position of a couple of residues, and what is the confidence in those relative positions? And PLDDT doesn't give that because by definition it is local. And so this is why we have the other metric, which is the predicted aligned error. So what you're doing is, if you had a true structure from experiment, let's say, you align a residue and then you find distance with all the other residues, and what is the error that you would get between the prediction and the true structure. It's not symmetric, this metric, but it's close enough. So if you had loops, and you can think about it, if you had structured part, it would be symmetric, but in the loops it may or may not be symmetric. So this is what this PAE does. So how would you use it? So let's take this example, which was all blue, which means PLDDT scores were really high, and see how would you use the PAE. So let's take two residues which are in the same domain, and this is the PAE plot that you would see on the website. For example, green is good, white is bad. Green means very low error in the relative position. White means the network thinks it's got it wrong, basically. It could be anywhere within 30 angstroms. And so if we look at these two residues, they are in the green region, which means the relative positions of these two residues are predicted with really high confidence. But if we look at the residues in the two domains, these two, for example, and plot them on this PAE plot, they are in the white region. And what this is telling me is that the prediction we have, where you see this two domains placed as they are in the coordinates, it's just a coincidence. They could be anywhere in space. And this is really important to make use of when you're looking at these structures. So this is just one example of how you could just lead down the path if you don't use these two prediction metric. So this is a exporting, and obviously the function is to export from the nucleus. Forget about the PLDDT and PAE for now. Look at the structure. It's a barrel with nice pore in the center. Obviously, you can start thinking about that pore has the function in terms of exporting. from the nucleus, but at the moment there is a helix there. And so you can start speculating about whether this has a functional relevance and whether this helix is kind of a cork and this is an inactive state and something happens and the helix comes out of this pore and it becomes active and all that. And then let's look at the metric, PLDDT and PAE. So the first thing to observe is that this particular helix, the PLDDTs are low, which means the confidence in the prediction of individual residues is low. But even more importantly, the PAE plot is telling us that the relative position of this particular helix, which is here at the N-terminus, with respect to the rest of the protein, is predicted with really higher, with very low confidence. And so it's just a coincidence that that particular helix is sitting in that pore. And it has no consequence, no functional relevance, and at least not based on this model. Right? So this model is not telling you anything about that particular helix. So I can't emphasize it enough. It's really, really important to use both PLDDT and PAE when you use these structures. Okay, so we're done with the method itself. Now let's see the developments that happen next and how that is going to impact life science research. So this is just a timeline of all the dates when we discussed certain important things and then launched the database. And as you can see, it was within about 3 to 4 months, which is quite an achievement, if I say it myself, because doing something like this requires discussions on many different things, starting from what we are going to put into the database, how we are going to organize it, and how we are going to display this information and all that. And some of it will come back in the slides that I have later. And then there were obviously more updates done. The focus was to basically increase the coverage of structured data in sequence space. And so we basically just added more and more structures. So there wasn't any development done on the website itself. It's more data. And so we now are up to 214 million structures in AlphaFold database. And as you will see in the slides, I think this is really one of the most important data sets that we now have after the human genome. So what are the limitations? So the database obviously carries all the limitations that the method has, and some of these are listed here. So the first is these are individual chains, individual protein sequences folded. They don't have their context. So most proteins work in complex with either small molecules or other macromolecules, and those are missing. And so that's important to consider. Having said that, you can look at examples and really be amazed. So this is just 2 examples. One is a homodimer, and you can see the prediction and the crystal structure exactly identical, including the loop that makes the interface in the homodimer. And the second one is even more scary and amazing. It's a DNA complex, and there is a loop that goes around the DNA, so it's hugging DNA like that. And even that is predicted perfectly in the AlphaFold database. And so the only thing missing there is the DNA. Everything else is really good. And these are anecdotal things, and I can find you another 10, but that doesn't mean every prediction is going to behave like this. And so that's another thing to remember, that just because you see one example doesn't mean that AlphaFold itself is able to do it repeatedly for all the other examples. The second thing, again, you see here examples of ligands, and we'll hear more on AlphaFill how this is useful in filling in the ligands. But you can see here a metal ion and all the side chains are more or less in the right place. There is a hole there where you can put in metal, and that's perfect. Similarly, this iron circle sulfur cluster, you can again see there is a hole there. Heme, you can see a hole there. But again, I can again tell you tens and hundreds of examples, and we'll see more from the AlphaFill talk. But that doesn't mean every one of those proteins is going to give you this type of information. So you have to be careful when you use a model that you want to use, and make sure that you're looking through it carefully. Right. So the next thing, there is no dynamics, and this has been. I have a slide where it's really becomes interesting. But in the database, there is no dynamics. AlphaFold doesn't predict dynamics. If you get many multiple structures from AlphaFold pipeline, again, I can show you examples where you can interpret them as dynamics, but then there will be hundreds more where you can't. So it's not predicting dynamics. If you get it, you are lucky if you validate it, but it's not predicting dynamics. As I said about intrinsic disorder, I won't repeat that. It's not trained to predict effects of mutations. There are papers which say if you mutate and predict, you get low PLDDTs, etc. Again, there are 5 examples in that paper, but it's not a characteristic of AlphaFold pipeline to predict an effect of mutations. In many cases, if you change one residue, you might get exactly same structure. So you have to be careful. You can obviously use your knowledge and other tools that are available in conjunction with AlphaFold model, like people have done with PDB models. But again, it's not AlphaFold which is doing it. So all this means that when you look at a model, it may or may not lead to a hypothesis. And if it does, well and good, you can design experiments and validate it. And we'll come back to this. How are we going to validate these structures? How are we going to make sure that this becomes a really useful resource for people who are not trained in structural biology? Okay, so where are we with AlphaFold database usage impact? I don't have to say much, lots of usage, lots of papers citing the method, lots of different applications in many different fields, etc. So let's just skip that, go to the next thing, which I think is the main impact in terms of structure coverage. And so when the database was launched, obviously we had 21 different proteomes covered, and the impact was that we went from about 17% residue coverage for human proteome to about 92% coverage. And that's important. So now we have models, which means these are all hypotheses. Now we can start designing experiments, start understanding biology. Don't have to wait necessarily for experimental structure, caveat, all the things I said before apply. And you have to be careful what and how you are using this. Why this is useful is because one of the things we knew about human proteome, or was predicted, was about 30 to 40% of the residues are disordered. And the models reflected that. And that's why there is at least one known fact or known information about human proteome is kind of reproduced through these models. So there is something there that we can start using and designing experiments. But in my opinion, the most important impact has been proteins are no longer seen as having a well-defined compact structure. And I think we got used to looking at really nice images and really structured things because of the experimental limitations of what was deposited to PDB. But proteins are not like that. Proteins have these disordered regions. And this, I think, is the biggest change in the mindset of almost everyone, I think, when they look at these structures. And this was a conscious decision. There was a big discussion about what should we put in this database. Should we just take the structured parts and put them in, or should we put the whole thing? And we made a decision that we put in everything for 2 reasons. One, because we wanted to distinguish these predictions from experimental structures. And this is one obvious way of doing that. And secondly, as it turns out, this is information, because in most cases, these disordered regions are disordered in the protein. And so it has helped, I think, change the way we think about proteins. And this is going to change, and is changing already, structural biology. So there are a couple of papers here. This is from yesterday, day before. But we have to remember, structural biology was never about having a structure. It was always about understanding function. And so now it's just becoming more evident that that's what we have to do for every biological problem that we apply structural biology to. And the predictions can help you in many different ways. So they can help in terms of designing the sample, the constructs, and obviously in solving structures as well. And have a look at some of these papers. They are really interesting. So you can almost have a pipeline now that, given a X-ray data, can just solve a structure in most cases. So everything is changing, how we look at structures and what we can do in terms of structural biology studies. And these are just 3 examples I have. So this is one where you take cell extract and you fractionate it and you put it on EM grid. The idea being that all the complexes that are not transient would fractionate and give you nice structures on EM grid. And that can be then assisted through these predictions as well to fit in these EM maps that you get. Really nice paper. You can see here. Nuclear pore, we have been trying to, so Jan Kosinski and others, Martin Beck, have been working on this for decades, and they had certain number of components put into this nuclear pore complex. And Jan basically started from scratch once AlphaFold came out, and within 3-4 months had doubled the number of components that he could fit into the EM map, and used actually the cross-linking to validate some of this as well. So really amazing work, and the speed at which we can now do these things is amazing as well. So we can start thinking about really in situ structural biology and what is called visual proteomics. In cell, looking at these molecules, different states of these molecules, fitting them at molecular atomic resolution using some of these predictions, etc. So really exciting time for structural biology. We can start understanding processes rather than individual structures. And that means it is obviously going to have impact on biological research as a whole. And we can now have protein structure basically for any protein you want and you want to study, and that's going to have impact. So there are many, many opportunities. First, just bioinformatics. So we're used to dealing with 200,000 structures. Now we have to deal with 200 million structures, with ESM4 now 800 million structures in total, and just the scale of it. And we need methods that can cope with that scale. So there is a huge opportunity to think about methods that are scalable and can work on this huge amount of data. Extending structure prediction, obviously complexes, protein ligand complexes, nucleic acid complexes, and there is a lot of work being done on that already. Addressing limitations, and I won't say much, you will hear more from Ida. Understanding dynamics, and I have a slide, and it's really interesting how we are thinking about it. And finally, we basically need to get to identify function of proteins, and how do we do that? So this is just the way things might change. The proteomics might become really important technique to get information that is sufficient to understand certain aspects of biological function. So for example, there are these 3 papers, one on just mapping all the PTM information on these predicted models and trying to get some new potentially regulatory sites, understanding that, and then design experiments to validate it, or chemical cross-linking and trying to understand which complexes exist and how they exist and things like that and their structure. So this might become really important technique. This or hydrogen-deuterium exchange, depending on what question we want to answer. And so it's really important for the proteomics community to develop the necessary data standards and archive this data so that we can use it in conjunction with these predicted models to validate them as well. This is a nice, I think, way of thinking about the whole process of how AlphaFold predicts a model. So this is a paper from Sergei Shnikov, and the idea here is you are looking at coevolution and seeding the initial kind of structure. What if you removed certain coevolution pairs? So if we think about it in experimental context, you have NOEs in an MR and you remove certain NOEs, and then you get a structure from the remaining data. The problem there is we don't know which NOE to remove, because if we remove one, which is the most important one, we might never get a structure that is meaningful. But this is not the case when we think about this, because what we are doing here, and this is where I want to highlight this paper, is we are taking the evolution, you're taking all the sequences, you're grouping them, and so in terms of constraints, if we think about it, evolution has taken care of giving you the right signals at different points in evolution. Obviously, this is not going to work for every protein we look at, but this is a really nice concept that I had not thought about or appreciated. The way you would do this is to then group these and get structures, and it might end up giving you information about the different states, depending on how evolution has modulated these things. And so really important kind of thought here, I think that can be explored a lot more. So what are the challenges with all this abundance of structures, about 800 million and counting, and maybe many billion now, with all the different labs predicting their own structures. So the first and foremost is fragmentation. We have all these different databases with all these predicted models. How can we make sure that those become accessible and we don't just keep predicting the same structures again and again? We also need common data standards so that they can be used by pipelines in the same way. And so that's one challenge. Lack of training, and I'll come to that. Validation, as I have mentioned, and making this useful. So how do we add the biological and medical context to these structures and make them interpretable for someone who is not really a structural biologist? Because at the moment, all this information is in publications. It's not in a parsable form in a database that we can easily use. So the first thing is data standards. So we have something called Model Safe, which is used by AlphaFold, RosettaFold, Model Portal, many different databases. And this is a standard that is used by PDB, the PDB XML SIF standard. This is an extension of it. And so you can add a lot more information to it, validate that that information actually relates to the data that you are providing. And so in terms of data standards, we have got our act together, we have something. How do we create a mechanism by which you provide access to all these different data sets? And so we started this project called 3D Beacons in 2019, anticipating that this is going to happen. Because AlphaFold 1 had already shown that predictions are becoming better and better. And so the idea here is we work with all the different resources that have data, and we have common APIs and we have common standards to make that data accessible. And so you can come to one place and search for a protein model. It goes to all the different data resources, brings back the models that you have. By definition, you get the quality of those models, etc. And so this is already there. Many different data resources are already part of this. So then let's get to training. So we now have access to data. How do we use it? So the first and foremost is who are the user communities? And then if we have non-structural biologists accessing this data. So for PDB, about 99% of our users are non-structural biologists. So I don't see why that would be different for these databases. And so how do you find structured data for your protein? How do you assess the quality? How do various structures differ if you have multiple structures? How to use these models to understand function? What are its limitations? A really important question. And so I think we need to really think about, as structural biologists, bioinformaticians, the training material that we need to produce to help all these users interpret these results, both from experiments and from predicted models. So this is just what happens when you don't have enough training, and so the challenges that we guard against our experimental data sets getting contaminated by predicted models put in without thinking in some experimental data that doesn't even support that kind of fitting of predicted model. So this is just one example. So as it says here, we have to guard against human silliness, although we have made strides in artificial intelligence. So that brings me to a really important point of validating all these things. So the way I look at it is we at the moment have a 3D tremble. So for those who don't know, UniProt is two parts: SwissProt, which is the manually curated data set, and Tremble, which is translated from gene sequences and then annotated automatically. And SwissProt is about probably 2-3%, and the rest is all Tremble. So what we have in AlphaFold database or ESN folder, whichever you take, is this Tremble. And we need to validate it, and we need to provide information in a way that these structures can be elevated to a SwissProt level, which means they become a lot more useful and a lot more reliable in terms of their use. So how do we do that? And this is where I think there is a big challenge, because it's no longer just the traditional methods that would do this. There will be methods like cross-linking, chemical footprinting, HDX, other methods, functional analysis, mutation analysis, biochemical experiments. All these add value. All these can be used to validate these protein models. And so how do we capture that? How do we say to a user that this whole model is validated, or just part of it is validated because we have a data set here which explains why this model, or this model explains that particular result. And so the challenge is, as a community, we have huge amount of data, and we need to work together, coordinate in getting all this information which validate these models together and make it accessible for people who can then use it in their experiments to understand biology, to understand biological processes. And this comes to then making this data useful, because we can see this is just a volume, maybe shape here. Once you make a trimer, even nicer shape, doesn't mean anything if you don't know what it is. You need to start adding extra information. Just a simple thing saying this is the spike glycoprotein gives you slightly more information. And then you start adding ligand binding site, known variants, etc. You are actually getting biology out of it, rather than just a three-dimensional shape and numbers in terms of atoms. So this is where we need to enrich these models and provide this structural and biological context. And we started this work on the PDB side in 2018 with PDB knowledge base. And so this is a community-driven resource where people deposit structures to PDB and they deposit annotations to PDB knowledge base. And we have about 33 different groups now in different countries. And then we have a way of displaying this data. Rather than individual structures, we need to start aggregating it to derive more biological insights. And that's what we try and do in KB, and maybe something similar needs to happen, and new visualizations need to be created as well. And we need to develop training material and make sure that anyone who is using these structures actually understands how to use them. So I'll just go through some of the important points. I think open access, so any data you have, make sure it is available in a way that can be used, which means use data standards, put it in the right database. The abundance of structure obviously is going to change everything. We need to create training, and this is going to have impact for decades to come, I think. And we can enhance that by really thinking about how we structure this, how we make the infrastructure available, and also innovative tools available for both the specialist and non-specialist users. So this is the last slide. I think AI is really fundamental new toolkit, and this is going on for some time. We have lots of biological data in genomics and now in structures, obviously, as well. And it's not necessary that we understand every possible detail. As long as we have good models with AI that give us some results that can be validated, it's a useful toolkit. And we can really understand biology and accelerate our understanding by using AI. And this is just training again. We need to up our game. Biology is seen as non-mathematician's field, which is not true, was never true. And I think we just need to address that. And this is something we are going to see in many, many different fields. So not just in structural biology, genomics, and every other possible field where we have lots of data. We are going to see some of these models come up. And so with that, I just want to acknowledge the team at PDBE, the team at DeepMind, and the funding as well. And I'll stop there. Thank you. Have questions?
So questions? Maybe two questions actually. So one is biological, one is more like dependency. First question, like equalization, how does it impact your model?
My name is Sally Chambers and I'm Digital Humanities Research Coordinator at Ghent University, and I also work at KBR, the Royal Library of Belgium, on digital cultural heritage material. Inspired by our colleagues at the Royal Library of Denmark, we're really keen to see how cultural heritage material can be computed using the supercomputer. For example, we have hundreds of years of digitized historical newspapers, but they're not necessarily machine readable or have been split into articles. The supercomputer can help, together with computer scientists, to train models in order to extract the knowledge that humanities researchers need. The VSC has been amazing in helping Claria, our humanities research infrastructure, in order to provide both access to the data, so for example, the digitized and born digital resources from the Royal Library of Belgium, and with facilities such as the Jupyter notebooks, can also help humanities researchers who are less computationally savvy to be able to still work with the digital cultural heritage data and do initial analysis and gain more confidence in being able to use supercomputing services. My dream is that we could have access to all European digital cultural heritage content on a supercomputer like the VSC, so that humanities researchers can analyze it. The VSC is helping us make that first step a reality.
Hello, I'm Álvaro Cortés and I'm bioinformatician at the Genomics Core Leuven. This is a shared facility between the University Hospital and the KU Leuven that facilitate the access of sequencing technology as well as supporting the data analysis and the data processing of DNA sequencing data. The purpose of the Genomics Core is twofold. We provide support for molecular diagnostic at the UZ Leuven and other hospitals in Flanders, and we support the sequencing workflows of research groups within the KU Leuven and other external groups. Before the VSC, we had to rely for both diagnostics and research on the internal cluster of the hospital. This posed unnecessary stress on our internal systems, as different projects were competing for limited computational resources. We then had to be very careful on how and when to schedule high-demanding computational jobs, as for instance de novo assemblies or whole genome analysis. Additionally, an increasing number of researchers wanted to analyze their data themselves, and they didn't have enough computational capacity to do it. With the VSC, we were able to move some of our complex research projects to the cluster at the VSC, alleviating the internal load of our own systems. We have successfully performed de novo assemblies, long-read iso-seq analysis, and RNA-seq analysis. We have also guided our research users on the use of the VSC to perform complex genomics analysis, which enable them to advance their science. The analysis of sequencing data is a demanding computational task. The VSC offers all computational capabilities to analyze such a data efficiently and with excellent assistance from the support team. If your research is in need of such analysis, the VSC would be my first recommendation.
My name is Wim Thierry and I'm a climate scientist working at the Vrije Universiteit Brussels. In our team at VUB, we use global climate models to study how climate extremes will evolve towards the future, and also to better understand the role that the land plays in shaping these climate extremes. So in our group at VUB, we use the Community Earth System model, which is a global climate model that represents all these climatic processes that occur in the atmosphere, the oceans, the land, and the ice sheets. Now, as a group, we contribute to improving the quality of that climate model, by introducing and improving the representation of water management and land management in that climate model. For instance, we incorporate reservoir management or irrigation techniques in that climate model. And by doing so, we improve the quality of the model, we make it more realistic, and this helps to generate more reliable future projections of climate change. So to run our climate model, we need access to high-performance computing infrastructure. And when we have that access, we can contribute climate simulations to the international climate science community. We can thereby support the development of the IPCC climate reports and also contribute to efforts such as the one shown behind me, where we quantify how many climate extremes a person born in a particular year in a particular country will face integrated across their lifetime. So doing this type of supercomputing simulations allows us to make international contributions to climate science. VSC is critical for performing our work as global climate modelers. With the help of VSC, we can run our global climate model on the supercomputer, and we can store the output on a shared data storage facility for further analysis. We are also collaborating with a whole range of other research groups in Flanders to share these resources for climate science analysis. And by doing so, we can improve our understanding of climate change, and we can help contributing towards solutions.
Today we are here at the Faculty of Veterinary Medicine of Ghent University. My name is Sebastiaan Teuns. I'm a veterinarian and co-founder of PathosSense, which is a new spin-off of Ghent University. With PathosSense, we offer complete diagnostics of infectious diseases for veterinary medicine. In this, we use nanopore sequencing, a revolutionizing way to analyze DNA and RNA of different kinds of organisms, including viruses and bacteria. Now today, Nick Verreke, my colleague, will take you along in his research in which he is combining nanopore sequencing and a lot of different computational analysis for which he uses the Flemish supercomputer.
We are working with nanopore sequencing. We started with this small MinION device, and recently we moved to the GridION, which allows us to generate a lot of more data. This sequencing data can be used for bacterial identification and antimicrobial resistance testing, which later on can be used for quick and real-time bacterial diagnostics in veterinary medicine. Could we have done this without the use of the HPC? Probably yes. While we started with a CPU-driven computer, we quickly went to a GPU-driven one. However, this did not sufficiently speed up our processes. So we went to the HPC, which could actually facilitate our programs and algorithms to speed up our analysis even further. VSC allows us to focus on the science and less on the computations, so we can improve our portfolio and the number of analyses within the spin-off company.
Thanks to the Flemish Supercomputer Center, we have been able to bring our research to the next level.
is Sally Chambers and I'm Digital Humanities Research Coordinator at Ghent University, and I also work at KBR, the Royal Library of Belgium, on digital cultural heritage material. Inspired by our colleagues at the Royal Library of Denmark, we're really keen to see how cultural heritage material can be computed using the supercomputer. For example, we have hundreds of years of digitized historical newspapers, but they're not necessarily machine readable or have been split into articles. The supercomputer can help, together with computer scientists, to train models in order to extract the knowledge that humanities researchers need. The VSC has been amazing in helping Claria, our humanities research infrastructure, in order to provide both access to the data, so for example, the digitized and born digital resources from the Royal Library of Belgium, and with facilities such as the Jupyter Notebooks, can also help humanities researchers who are less computationally savvy to be able to still work with the digital cultural heritage data and do initial analysis and gain more confidence in being able to use supercomputing services. My dream is that we could have access to all European digital cultural heritage content on a supercomputer like the VSC, so that humanities researchers can analyze it. The VSC is helping us make that first step a reality.
Hello, I'm Álvaro Cortés and I'm bioinformatician at the Genomics Core Leuven. This is a shared facility between the University Hospital and the KU Leuven that facilitate the access of sequencing technology as well as supporting the data analysis and the data processing of DNA sequencing data. The purpose of the Genomics Core is twofold. We provide support for molecular diagnostic at the UZ Leuven and other hospitals in Flanders, and we support the sequencing workflows of research groups within the KU Leuven and other external groups. Before the VSC, we had to rely for both diagnostics and research on the internal cluster of the hospital. This posed unnecessary stress on our internal systems, as different projects were competing for limited computational resources. We then had to be very careful on how and when to schedule high-demanding computational jobs, as for instance de novo assemblies or whole genome analysis. Additionally, an increasing number of researchers wanted to analyze their data themselves, and they didn't have enough computational capacity to do it. With the VSC, we were able to move some of our complex research projects to the cluster at the VSC, alleviating the internal load of our own systems. We have successfully performed de novo assemblies, long-read iso-seq analysis, and RNA-seq analysis. We have also guided our research users on the use of the VSC to perform complex genomics analysis, which enable them to advance their science. The analysis of sequencing data is a demanding computational task. The VSC offers all computational capabilities to analyze such a data efficiently and with excellent assistance from the support team. If your research is in need of such analysis, the VSC would be my first recommendation.
My name is Wim and I'm a climate scientist working at the Vrije Universiteit Brussels. In our team at VUB, we use global climate models to study how climate extremes will evolve towards the future, and also to better understand the role that the land plays in shaping these climate extremes. So in our group at VUB, we use the Community Earth System model, which is a global climate model that represents all these climatic processes that occur in the atmosphere, the oceans, the land, and the ice sheets. Now, as a group, we contribute to improving the quality of that climate model, by introducing and improving the representation of water management and land management in that climate model. For instance, we incorporate reservoir management or irrigation techniques in that climate model. And by doing so, we improve the quality of the model, we make it more realistic, and this helps to generate more reliable future projections of climate change. So to run our climate model, we need access to high-performance computing infrastructure. And when we have that access, we can contribute climate simulations to the international climate science community. We can thereby support the development of the IPCC climate reports and also contribute to efforts such as the one shown behind me, where we quantify how many climate extremes a person born in a particular year in a particular country will face integrated across their lifetime. So doing this type of supercomputing simulations allows us to make international contributions to climate science. VSC is critical for performing our work as global climate modelers. With the help of VSC, we can run our global climate model on the supercomputer, and we can store the output on a shared data storage facility for further analysis. We are also collaborating with a whole range of other research groups in Flanders to share these resources for climate science analysis. And by doing so, we can improve our understanding of climate change, and we can help contributing towards solutions.
Today we are here at the Faculty of Veterinary Medicine of Ghent University. My name is Sebastian Deuns. I'm a veterinarian and co-founder of Tassos, which is a new spin-off of Ghent University. With Tassos, we offer complete diagnostics of infectious diseases for veterinary medicine. In this, we use nanopore sequencing, a revolutionizing way to analyze DNA and RNA of different kinds of organisms, including viruses and bacteria. Now today, Nick Verreke, my colleague, will take you along in his research, in which he is combining nanopore sequencing and a lot of different computational analysis for which he uses the Flemish supercomputer.
We are working with nanopore sequencing. We started with this small MinION device, and recently we moved to the GridION, which allows us to generate a lot of more data. This sequencing data can be used for bacterial identification and antimicrobial resistance testing, which later on can be used for quick and real-time bacterial diagnostics in veterinary medicine. Could we have done this without the use of the HPC? Probably yes. While we started with a CPU-driven computer, we quickly went to a GPU-driven one. However, this did not sufficiently speed up our processes. So we went to the HPC, which could actually facilitate our programs and algorithms to speed up our analysis even further. VSC allows us to focus on the science and less on the computations, so we can improve our portfolio and the number of analyses within the spin-off company.
Thanks to the Flemish Supercomputer Center, we have been able to bring our research to the next level.
My name is Sally Chambers and I'm Digital Humanities Research Coordinator at Ghent University, and I also work at KBR, the Royal Library of Belgium, on digital cultural heritage material. Inspired by our colleagues at the Royal Library of Denmark, we're really keen to see how cultural heritage material can be computed using the supercomputer. For example, we have hundreds of years of digitized historical newspapers, but they're not necessarily machine readable or have been split into articles. The supercomputer can help, together with computer scientists, to train models in order to extract the knowledge that humanities researchers need. The VSC has been amazing in helping Claria, our humanities research infrastructure, in order to provide both access to the data, so for example, the digitized and born digital resources from the Royal Library of Belgium, and with facilities such as the Jupyter notebooks, can also help humanities researchers who are less computationally savvy to be able to still work with the digital cultural heritage data and do initial analysis and gain more confidence in being able to use supercomputing services. My dream is that we could have access to all European digital cultural heritage content on a supercomputer like the VSC, so that humanities researchers can analyze it. The VSC is helping us make that first step a reality.
Hello, I'm Álvaro Cortés and I'm bioinformatician at the Genomics Core Leuven. This is a shared facility between the University Hospital and the KU Leuven that facilitate the access of sequencing technology as well as supporting the data analysis and the data processing of DNA sequencing data. The purpose of the Genomics Core is twofold. We provide support for molecular diagnostic at the UZ Leuven and other hospitals in Flanders, and we support the sequencing workflows of research groups within the KU Leuven and other external groups. Before the VSC, we had to rely for both diagnostics and research on the internal cluster of the hospital. This posed unnecessary stress on our internal systems, as different projects were competing for limited computational resources. We then had to be very careful on how and when to schedule high-demanding computational jobs, as for instance de novo assemblies or whole genome analysis. Additionally, an increasing number of researchers wanted to analyze their data themselves, and they didn't have enough computational capacity to do it. With the VSC, we were able to move some of our complex research projects to the cluster at the VSC, alleviating the internal load of our own systems. We have successfully performed de novo assemblies, long-read iso-seq analysis, and RNA-seq analysis. We have also guided our research users on the use of the VSC to perform complex genomics analysis, which enable them to advance their science. The analysis of sequencing data is a demanding computational task. The VSC offers all computational capabilities to analyze such a data efficiently and with excellent assistance from the support team. If your research is in need of such analysis, the VSC would be my first recommendation.
My name is Wim and I'm a climate scientist working at the Vrije Universiteit Brussel. In our team at VUB, we use global climate models to study how climate extremes will evolve towards the future, and also to better understand the role that the land plays in shaping these climate extremes. So in our group at VUB, we use the Community Earth System model, which is a global climate model that represents all these climatic processes that occur in the atmosphere, the oceans, the land, and the ice sheets. Now, as a group, we contribute to improving the quality of that climate model, by introducing and improving the representation of water management and land management in that climate model. For instance, we incorporate reservoir management or irrigation techniques in that climate model. And by doing so, we improve the quality of the model, we make it more realistic, and this helps to generate more reliable future projections of climate change. So to run our climate model, we need access to high-performance computing infrastructure. And when we have that access, we can contribute climate simulations to the international climate science community. We can thereby support the development of the IPCC climate reports and also contribute to efforts such as the one shown behind me, where we quantify how many climate extremes a person born in a particular year in a particular country will face integrated across their lifetime. So doing this type of supercomputing simulations allows us to make
to bring her here, but without further ado, I will introduce you Janani Durairaj. Structure is the new sequence.
Okay, so I can start. Hey everyone, I'm Jade, and I hope you can all hear me and see my slides. I was supposed to be there in person, but I had a lot of travel adventures yesterday, so I couldn't make it. Still happy to be giving this virtually. Okay, so the title of my talk is Structure is the New Sequence, and it seems a bit controversial, but I think you'll see by the end that I don't really mean what it seems like I mean. So everything is working, right? Okay, I'm gonna assume that things are working. The reason that I can even make such a bold claim is mostly because of AlphaFold and things like it, is that now we have number of structures is almost similar now to the order of magnitude of number of sequences. And since structure is in some sense one step closer to function, we can learn a lot more theoretically from having structure in addition to having sequence about protein function. But what I really want to talk about is not exactly that, but about from a bioinformatics or computational biology perspective, about the algorithms involved. Because we have a lot of algorithms for protein sequences. We've had them for decades. We have some new ones over the past years. They've been highly optimized. They've gone through a lot of improvements, and I think a lot of people in the field are really familiar with sequence algorithms. And so I really want to talk about how that is now translating to the structures or the huge influx of structures that we now have. And so for this, I want to talk about three different classes, let's say, of methods. K-mer counting, which is when you fragment your protein sequence into overlapping K-mers and then see what kind of K-mers are available. This is the basis of a lot of different algorithms that build on top of this. And then alignment, so you can have pairwise alignment, you can have multiple sequence alignment. And then this is, of course, the basis for things like phylogenetic trees and so on. And finally, prediction or machine learning, which is quite vague, of course, but it covers this idea of having your sequences, extracting features from them, or creating a model that extracts features from them, and then using that to make some insights on either the data that you're already training on, which is unsupervised, or on some new data. And so for all these three, I want to give some examples of how structures are now being used in similar situations. I'll talk about stuff that I've been working on, but also about what other people have been doing in these areas. So starting with K-mer counting or K-merization. So this works really well for sequences, of course, because there are 20 discrete amino acids or nucleotides or whatever, and you can really easily split your sequence and then count the number of times different subsequences appear. How would you do something like this for structures? So first of all, you can fragment your protein structure in a similar way. You could look at overlapping coordinates in the backbone, or you could look at overlapping radii that have some atoms within them. But of course, these are all in three-dimensional space, so they're rotated and translated, so you can't really say this one is equal to this one, like you could with amino acids. And so for this, I looked into rotation invariant moments. So this is a concept from the field of computer vision, where they use it for things like characters recognition and pose recognition and so on. Essentially, it's a mathematical formula that you feed in a set of coordinates over here through this formula, and it gives you a number. But if you rotate these coordinates and feed it through the same formula, you get the same number. And so here's an example with just this bunny. It has a lot of points, and you can rotate it in many different ways. But you'll see that these are 4 moment invariant formulas. They all have exactly the same value for all of these different rotations. And so what will be interesting, of course, is to use this in a search sort of situation, or in a way of actually describing fragments. So here I have the example of these 2 ears from the bunny, which you would expect, of course, to be similar, rotated and translated in different ways, and then those same 4 moments for those 2 ears in blue and in orange, and then for the random parts of the rest of the bunny of the same size. And you can see that the ears are kind of closer to each other, meaning that not only do these moment invariant formula give you the same number when you rotate the coordinates, but you can use them to somehow describe different shapes. And so this is the concept that I used for bringing k-merization to the structure world, and it's what we call shape-mers. And essentially, each shape-mer represents C-alpha coordinates of 16 residues in the backbone and 10 angstroms around it. So not just the backbone, but also interactions with neighboring chains. And so I have now 1,024 of these. Just to give you an idea of what these look like. Also, this is of course not a new idea to fragment proteins into protein structures into shapes. I think what's sort of, let's say, new here, or not so common here, is that it's not just the backbone anymore, but also what comes around it. And that becomes really important. So here, let me just explain what this shows. So this is a representation of shaper on the left, and then on the right are some examples of proteins, which are mostly made up of the shaper, so repeat proteins, which, just to show you how it looks like when it stacks up. And you can see that because we have this radius information along with the backbone information, you can now distinguish something like this, which is also very difficult, but in a very different way from something like this. So it captures both short and long range information. And so that's what we call geometricus, and this is what we're now using for doing a lot of things that people do with sequence gamers on sequences, now with structures. Okay, so that was about gamers. The next thing that I want to talk about is about alignment, which of course forms the basis for a huge number of sequence algorithms. And I think many of you may have heard already of FoldSeq. So this is structure alignment, structure pairwise alignment, and this actually does make use of a lot of sequence-based approaches, right? So FoldSeq converts structures into a sort of sequence, where instead of amino acids, you have 20 characters representing some kind of 3D state. And then uses techniques in MM6, which uses k-mer matching and then sequence alignment to then perform structure alignment. So another example where we're now reusing the algorithms that we've somewhat honed for sequences to structures. And so this is for pairwise alignment, and I think this is great because it's very fast. It's almost as fast as sequence-based methods, but uses structures. And then we want to do the same thing for multiple structure alignment. And so there are existing, of course, multiple structure alignment tools. These are not optimized like sequence alignment tools, usually. And they tend to have this problem of creating very gappy sort of alignments. So we wanted to address both of these things. So first of all, the main time-consuming step in multiple structure alignment is deciding which two pairs of which pair of proteins to align at each step. Because you start with your whole set of proteins, you choose a pair to align, you align them, and then you start adding to that alignment. And so choosing that usually requires all versus all alignment, which takes a lot of time. So we've now replaced that with all versus all shape counting, which is what is done in a lot of multiple sequence alignment methods like cluster and so on. And we've also added some, let's say, feedback loop in the alignment process so that regions which are well aligned, you don't add too many gaps there. And that is really good for if you want to use your alignment as an input for machine learning tasks, because then if you have a situation, for instance, like this, where you have maybe two distinct clusters of structure in certain regions, you would still want to compare the features in between these. Yeah, so this is what we call Careta. It's quite fast. Still, of course, can be improved. I mean, I think multiple sequence aligners can be much faster than this. But there are a lot of tricks still left in the sequence alignment field that can get up to structural alignment here. Okay, and then I'll spend most of the time talking about this very vague concept of machine learning. Because, yeah, there is a lot to be said here. And so, as I said, when you have protein sequences, what you would normally do is you have your set of sequences, you find a way to extract some representation from them. It could be actual features, like physical chemical properties and so on, or it could be that you put them through a language model and then you get a different kind of representation and so on. And then you would use that either for just similarity maybe, or for dimensionality reduction, you could use it for clustering, or you could use it for making prediction of function or mutation effects and so on. And now you can finally, to some sense, do something similar with structure at the same scale. And we're seeing a lot of this happening now with a lot of recent papers. And so it's the same basic idea. You have your set of structures, you would extract maybe features or use another black box techniques to obtain your representation, and then train your model to perform the task measures. And of course, what's really interesting in machine learning is then to map back the insights that you find to the structure itself, maybe in terms of these residues are predictive for a certain thing, or these regions are responsible for this, and so on. And here I want to make this distinction between protein family and protein universe, because when you are working on a task inside a particular protein family, let's say you want to predict kinase activation or something like this, or ligand specificity in a certain enzyme class, then you would be using quite different techniques than if you're looking at clustering the entire protein universe. First of all, in the protein family case, you're much more likely to use multiple sequence alignment or alignment-based techniques because there the residue correspondence between different proteins is meaningful because they represent quite similar structures with some variation. Whereas in the protein universe case, you would use things like shapers or something that's much more broad because you can't have this meaningful residue correspondence across all the proteins in the protein universe. And similarly, the kinds of, let's say, subunits that you use in machine learning changes as well. Like you could use the entire structure, or you could be looking at a residue-based level, especially for things like mutant prediction. You would probably look at features of a single residue or features of a single residue along with its local environment. Okay, so that's let's say the general idea, and now I'm going to talk about some specific examples. I'll start with stuff that I did in my PhD in the Netherlands, and here I was looking at a class of enzymes called terpene synthases. And you don't really need to know so much about them, it's just that these are plant enzymes that make essentially all the fragrances of plants, so the smell of orange, the smell of mint, etc. And what was interesting about them is that they their sequence varies a lot. So they can have as low as 20 to 30% sequence identity and make exactly the same product, or they could have as high as like 99% sequence identities for mutations and make completely different products. So this was really an area where structure-based approaches were almost necessary, I would say. And so for this, I could use quite a classic machine learning approach where at that point it was homology modeling and not AlphaFold, but then you extract different kinds of features from the structure and then train a machine learning model to predict what kinds of products are being made. And then you can inspect that model to then say which residues could be involved in the creation of certain products. And this was quite successful. We had some nice results, and we were able to look at some really interesting residues. And then I'm now following up on that to extend this to all the 300 or so products. And this is where you then need also features from the ligand perspective. There are a lot of papers on this as well coming out now where you have structural features from the protein and then maybe structure or 2D features from your ligand, and then you predict whether those two are compatible, let's say, if they bind or not. And then you can use a lot more, you can use interpretation methods to then pinpoint residues that may be involved in the creation of individual products. Okay. Then I want to talk about some recent work that I've been doing with Joanna in our group in Basel. And what Joanna is trying to do is really map the darkness or the unannotated protein universe. So if you look across the protein universe, you have annotations from Swiss prot, from Interpro. People have done a lot of experiments and so on. So you have proteins which are really well described, you know what they do. You have proteins where you may know what they do and so on. And then you have proteins which are similar to the ones where you know what they do, so you also kind of know what they do. But then there are proteins which are dark, where nobody really knows what they do. There haven't been experiments done, and these are of course really interesting because what do they do? So what she's done is essentially map across UniProt, UniPark, and UniRef all the Interpro annotations. And then if you look at UniRef 50, which clusters protein sequences at the level of 50% sequence identity, you can estimate how much is known about each UniRef 50 cluster. And then the assumption, of course, is that if you know something about something within the UniRef 50 cluster, you probably know about the rest of its members as well. And so that way you get, let's say, the dark UniRef 50 clusters. And then what she did was to link these all together, all the UniRef 50 representatives, just based on sequence identity. So with the huge MMSeq search, all 25 million or so against all MMSeqs, with 50% coverage as well, so that you know that it's at least covering half of the sequence. And then here she mapped the darkness into this huge network and then tried to see, are there really dark, let's say, clusters in here that we don't know anything about? And so in some cases, of course, it's that even though it's dark, it's connected to proteins where we do have a lot of information, so it's not, but we could theoretically annotate these kinds of sequences. And then there are the ones which are more distributive, which we do have information about, but then it's not at the level that UniProt would say, okay, exactly. But then there are quite a few actually clusters where we really have no idea. And this is something that Joanna is quite used to doing with these let's say, targeted evolutionary analysis. And so I just want to show you an example of how she went through this now with these structures available. So we have this cluster and we want to find out what it does. And so what she usually does is, of course, a search. So the reciprocal investigative search to find homologs and parallels. And then with this, from this, she found that, it's present in a lot of bacteria. And now here is where something new comes in, is this new tool called Vries, which is a structure-based protein function predictor. And essentially it uses language models and from the sequence and then the structure to at a residue level predict different kinds of filters. So for instance here you could say that this area is predicted to be calcium ion. And so now she runs this as well on these dark proteins that she's trying to find, and then it predicts DNA binding in some regions and hydrolase activity in some other regions. And then now we go back to sequence, because this is the next step that she usually does, is look at the genomic context, so to see what's next to it in the operon. And then this, the one that's next to it, also turned out to be dark and non-annotated. But again, we now have the structure of this as well. And so you can do the same kind of prediction there, and then you have much more of a hypothesis than you could have probably before. So this is just a really, I thought, a nice example of how structure is kind of changing quite routine analyses that people have been doing. Okay, so then that was, let's say, protein family perspective. Now what can we say about protein universe from a structure point of view? So can we learn more about proteins by looking across the entire AlphaFold database? And how does it differ from the PDB? Like, what are we, what have we gained in compared to the PDB, or what does the PDB have more than AlphaFold for some reason? And does it predict structures which are completely different from what we've seen before? And so to do this analysis, I focused only on the high confidence AlphaFold structure, so more than 90 average POPT, and high confidence regions as well. So I did not look at shapers from these loopy regions, which would also not make sense from a shaper point of view, since they're not really structured. But of course, those parts could be quite important as well, as we heard from the previous talk. So this is just a disclaimer that we look at only the structured regions for these analyses. And then I applied a bunch of natural language processing techniques. So shapers are similar to k-mers, as we saw before, but they're also similar to words and documents. So now you can use these algorithms that were made for natural language on protein structures. So two kinds of things. First is topic modeling, which, if you think of it from document point of view, is if you have a set of articles, you want to find out what kinds of topics these articles are talking about, and you do that in a completely unsupervised way. So you look for combinations of words that appear together more often. And so if you do the same thing for protein structures, essentially you come up with combinations of shapers that appear more often together across different proteins. And then from these topics, I want to see how is it different in the AlphaFold DB compared to the PDB. And so yeah, I think this is what I'm aiming for, is that the PDB gives a skewed view of natural proteins. And this we sort of know somehow to be true, because of course people crystallize things that they are interested in studying. Some things are not so easy to crystallize, and so there is some bias in terms of what kinds of structures are present in PDB. And then the second step is outlier detection. So here I essentially make an embedding for the whole protein, and then see if AlphaFold proteins are, let's say, structural outliers compared to PDB proteins. And here by outlier, I just mean that they have an unusual combination of shapers. Okay, so let's look at the topics first. This is some initial results from this. So first off, let's talk about what AlphaFold has more than. So of course, since AlphaFold has 200 million structures, everything is more than PDB. So that's not really the question here. It's like proportionally, if we took the PDB distribution of proteins as, let's say, natural, and extended that to what we would expect to find in AlphaFold, do we find similar sort of distributions? And if we don't, then which ones do we find more in AlphaFold and which ones do we find more in PDB? And so what I found is that AlphaFold has way more of these alpha beta barrel kind of enzymes. Also, I think this is quite known, is that it has a lot more transmembrane proteins. Transmembrane proteins were not so easy to crystallize. AlphaFold has a lot of green valence as well. And it has more repeats of, like, for instance, plant pentacyclopeptide proteins. You have crystal structures of some parts of it, but then we have much longer repeats of that. And then from the PDB perspective, it has more antibodies. I think this is also sort of expected because we have crystallized antibodies from a lot of different studies and a lot of different conditions and so on, often on the same sequence. And now this is actually, let's say, a side effect of the technique. The PDB is seen as having more disordered loops because, of course, we remove disordered loops in the AlphaFold structures. Yeah, so these are the kinds of, let's say, things that we could maybe learn from a larger scale. For instance, another thing that I found is that PDB has more representatives of ribosomal proteins. And this could be, I'm not sure, I have to check this out, but it could be because ribosomal proteins generally form huge complexes. And maybe AlphaFold is not able to reproduce the exact same number that's found in these huge complexes. Okay, and then if we look at the outliers and cross that with darkness that I was talking about before, first off, like, let me explain what the figure is. Each dot is a protein. It's connected based on the structure similarity, and the color is the darkness. So if it's dark, then it's not very well annotated, and if it's bright, then it's a well annotated protein. And then the size is the outlier, is whether it's an outlier or an inlier according to our outlier analysis. And so you have cases like this where everything is an inlier, and actually based on structure similarity, you can see that these proteins are quite similar. Probably you could easily figure out what this protein does based on looking at what that protein does. And then you have cases like this where you have, let's say, partial, in this case, it's almost like partial beta barrel. And if you do a structural alignment, then it matches really well with the beta barrel, but of course it's not complete. And that's why it's a structural outlier. Of course, the combination is not what you see so often in PDB. And this is actually a quite interesting example because it's turned out to be, I don't know if it's this one exactly, but one of the ones in here, which is like this partial beta barrel, is like an antimicrobial peptide that is known to interfere with gram-negative bacteria. So now we can maybe make the hypothesis that it interferes with their beta barrels, and this structure that it has could somehow be related to that. So we're trying to follow up on this with some experimental studies now. And then the last thing is that you can have a lot of, let's say, quite rare things that are quite rare in PDB. So there are, of course, some PDB representatives in here, but then it turns out that maybe there are a lot more than what we would expect forming these kinds of structures. And in general, the outlier detector also detects a lot of repeat proteins, and this you can see is because it's not so common, of course, to have long repeated secondary structure elements in the PDB, so you would have more globular proteins. So this is sort of a detector of, could also be seen as a detector of whether it is a real protein or not. So this is also something that we're trying to look into. And at the same time, there are some which are really, it just was kind of impossible to crystallize. So for example, this one here, there's a paper about this now, is that some people had not been able to crystallize this because it was hard to express and purify, and now we have the structure which seems to match what people expected that the structure would be. And then I thought this one was interesting because it's, yeah, you can see that it looks quite weird, but this is over 95 PLDT on average, so all of this is high confidence. So this is something that I just want to look into and see how is that happening. And then there are cases like this where you do have a high structure hit, but then there, in some regions, the topology is quite different from what you may expect. Okay, so that's sort of, let's say, we're going to continue from here and sort of going through all these different avenues that I just talked about. And so this is sort of a protein universe view of finding interesting places to look for new functions or new folds or things like this. So that was what I wanted to say, and essentially this is where we end up. So what I meant by structured new sequence is, can we use all the same algorithms and methods and ideas that we have for sequence, can we now apply that to structures? And it's not just structures actually, because I mean, anything that we can represent as a sequence of residues, it could be amino acids, it could be 3D coordinates, but it could also be, for instance, the language model representations that are starting to pop up now. And all of these could be used in these tasks that we are aware of and used to doing, which is looking at clustering, similarity, using alignment, and then using that alignment to make phylogenetic trees, looking for different kinds of motifs and patterns, and finally for function prediction. And I just want to leave you with this, which are some recent ideas along these directions where you would expect the approach, like you probably have heard the approach from a sequence perspective, and now it's being used from a structure perspective. Or from a language model perspective in this case. Yeah, okay, so thanks a lot. I'm not sure exactly how to answer questions.
So we'll try. Can you see me actually? Janani, can you hear me? Janani, can you hear me? I think we need to turn her volume up on her side. Janani? Maybe send her a...
Yes.
All right. All right, we're doing it with your phone. Is it possible to turn on your audio on your... You can't hear me, I guess, from here.
No, I can hear you. So, you want...
Can you hear us? Not without the phone. No, you can. Okay, you cannot hear us. All right. So I will try to ask here. Do we have any questions from the audience? I guess we need to be answering via the phone. Please go ahead. Really, it's better for this. It'll be translated. That's.
is Sally Chambers and I'm Digital Humanities Research Coordinator at Ghent University, and I also work at KBR, the Royal Library of Belgium, on digital cultural heritage material. Inspired by our colleagues at the Royal Library of Denmark, we're really keen to see how cultural heritage material can be computed using the supercomputer. For example, we have hundreds of years of digitized historical newspapers, but they're not necessarily machine readable or have been split into articles. The supercomputer can help, together with computer scientists, to train models in order to extract the knowledge that humanities researchers need. The VSC has been amazing in helping Claria, our humanities research infrastructure, in order to provide both access to the data, so for example, the digitized and born digital resources from the Royal Library of Belgium, and with facilities such as the Jupyter notebooks, can also help humanities researchers who are less computationally savvy to be able to still work with the digital cultural heritage data and do initial analysis and gain more confidence in being able to use supercomputing services. My dream is that we could have access to all European digital cultural heritage content on a supercomputer like the VSC, so that humanities researchers can analyze it. The VSC is helping us make that first step a reality.
Hello, I'm Álvaro Cortés and I'm bioinformatician at the Genomics Core Leuven. This is a shared facility between the University Hospital and the KU Leuven that facilitate the access of sequencing technology as well as supporting the data analysis and the data processing of DNA sequencing data. The purpose of the Genomics Core is twofold. We provide support for molecular diagnostic at the UZ Leuven and other hospitals in Flanders, and we support the sequencing workflows of research groups within the KU Leuven and other external groups. Before the VSC, we had to rely for both diagnostics and research on the internal cluster of the hospital. This posed unnecessary stress on our internal systems, as different projects were competing for limited computational resources. We then had to be very careful on how and when to schedule high-demanding computational jobs, as for instance the de novo assemblies or whole genome analysis. Additionally, an increasing number of researchers wanted to analyze their data themselves, and they didn't have enough computational capacity to do it. With the VSC, we were able to move some of our complex research projects to the cluster at the VSC, alleviating the internal load of our own systems. We have successfully performed de novo assemblies, long-read iso-seq analysis, and RNA-seq analysis. We have also guided our research users on the use of the VSC to perform complex genomics analysis, which enable them to advance the use of the VSC.
The analysis of sequencing data is a demanding computational task. The VSC offers all computational capabilities to analyze such a data computationally and with excellent data from a single use of the VSC. If you reach a complex genomics analysis with enable them to apply the use of the VSC to perform complex genomics analysis, which enable them to apply the use of the VSC to perform complex genomics analysis, which enable them to apply the use of the VSC to perform complex genomics analysis, which enable them to apply the use of the VSC to perform complex genome, and now we have access to over 200 million predicted protein structures that we can all access through the AlphaFold database that's made to different analysis. And we can use them to design our experiments. However, those protein models are, those AlphaFold models are protein only. For proteins, they are not stable and they can also not function without compounds that are crucial for structure protein. There are compounds that are important for structural integrity, for maintaining or getting to a certain fold. Compounds in fold here are metal ions, but also bigger organic molecules. For instance, if we look at this protein, which is myoglobiin C, then AlphaFold predicts the fold of the protein properly. But the information about the function of this protein is not in there. We do see that there is a secretary for a group which myoglobin needs to bind oxygen and carbon monoxide to be able to transport those two molecules throughout the body. The histidines are there, they are ready to bind the heme, but the molecule itself is not there, neither are oxygen molecules. And there are also compounds that are important for protein function, organic ligands and cofactors. And cofactors usually are important for enzymes to be able to do their function, perform the enzymatic reactions. For instance, this is adenosine-methionine in metal transferases, so it's important to transfer a metal group from one molecule to another, but also cofactors and and this made us wonder, can we enrich the AlphaFold models until the structure models that we have available in our Event. And this is a database that we have developed for almost 50 years now, and it contains really fine rebuilt and uniformly treated picture structure models. So we went on. One of the questions that we first would like to answer is which compounds we like to transfer. Obviously, we would like to transfer heme into myoglobin, but also other compounds like all the cofactors. There's a cofactor database with all the protein compounds to get commonly occurring ligands from the PDB, for instance HB. And the PDB also contains some molecules that are used for the experiments to, for instance, give a long protein density structure. And then we map them back. So when we find A and P, we map back to it. And we also select commonly occurring metal ions. We decided to not transplant compounds like water and other chemicals to save proteins because those are usually used to facilitate the experiments rather than protein. Also, we did not select post-translational modifications or amino acids because such modifications usually involve a conformational change in the structure that we don't know how the states of the AlphaFold models. And the same holds for other compounds like peptides and amino acids. So when we do this, we look at the PDB and we selected all the compounds that have 95% coverage of full alleles present in there. And this we sorted into the transplantable compounds list with about 2,000 compounds. And with that, we could start developing our algorithm. So we started with an AlphaFold model initially, and nowadays we also use an we first do a blast sequence search against all the sequences that we have in Event. And we look for a minimal sequence identity of 25% of the length 85 amino acids. And this is because that is the minimum requirement to get the secondary structure identity to Event. And with this case, we check if there are widths that have a transplantable compound. one step left, and that is actually to copy the transplant the transplant from the video reader model into the AlphaFold model, and then we have the AlphaFill models. We do make sure that we transplant or you can copy other certain compounds. So we apply this algorithm to the AlphaFold database version 2, so that is almost 1 million predicted protein structures, and it contains open body organism proteins, relevant to global health and analysis. When we do this, we end up with 586,000 AlphaFold models with one transplant, and in total we have 10 transplants over 12 million compounds. If we translate into model-driven, yes we did. You can see it on the right-hand side, so this is the AlphaFill model where it is transplanted as well as transcript molecules the more recent AlphaFold models, so remaining over 200 million, can be generated on the fly, and then we will switch. The algorithm is fast enough to do that in usually a couple of seconds. Slightly we can do that, maybe we need some validation. In order to do so, we created a condition set, which contains all the transplant compounds that originated from the homogeneous and for those transplants, we calculate the local environment condition score, the X score. And this is the RMSD that we calculated for all the non-hydrogen atoms within the six-dimensional block of the VSC. And you can see the distribution here with a p-value 0.5, so that's distribution. However, we can only calculate the score for transplant that originated from the VSC. So we cannot use it as is to define some quality metrics for transplants. That is why we started our score with VSC over to consider parameters, so we can square root of the sum of the squares from the box overlaps divided by the number of the squares. And we consider all the distances within 4x. When we again correlate this score, when we correlate this score with validation and the X score, also here we observe a correlation. And therefore we decided that we can use this TCR to class score to indicate transplant compatibility and quality. So that is for the technical part. That's where I will focus. As you know, the show this morning, AlphaFold is in that network of compliance structural database. But we also have our web service. This is the entry page where you can access the unified ID of your protein details. And then if you press enter on the keyboard to update your notes. So first of all, here in the top you can see that you can also go to homepage or have and when you go to structures you get a list of structures with the number of transplants, other details. When you go to compounds you get a list of compounds with the numbers to which transplant represents. So you get some statistics. And now we're looking at the methods. So we can see that we're looking at the average called transplant and you can download the structure file, which is the AlphaFill model, the metadata, which is very important annotations, and if you read it as well, and the link to the original AlphaFold. Then on the left-hand side, you can see the 3D representation of the protein, and this is interactive, so you can toggle it around, zoom in, zoom out, depending on the protein, and you can migrate over that. For this protein, we see that the transplanted compounds at the bottom are mainly hydrophobic, and this indicates that this is the transmembrane part of the protein. And on the more bottom part, the subdomain of the protein, we see that the lipids are more correlated with the protein. This is already an indication that you would not necessarily get from the
Then we have some scores, the global RMSD, just to give you an indication about the global alignment of those two structures, an ASIM-ID, so you can find back which one is which. And then the two quality indicators that I introduced you before, the local RMSD and the TCS, which is the clash score. And you can see that some of those scores are marked yellow. And this indicates that you should be a bit suspicious by using those transplants, because those scores indicate that there might be something not that proper. If those scores would be really worrisome, then those blocks would be marked red. So it's rather intuitive how much caution you should take with the transplant based on those validation metrics. Then finally, we have the show column where you can select the tick boxes of certain ligands you want to have a look at. And then there is, since yesterday, the download button that you can also download the thing that you're exactly the thing that you're looking at in the 3D viewer at that point. When you only select one ligand, we can, as you can see here for this cellular retinoic acid binding protein 2, we have selected the one on the bottom, which is retinol. It has a class score of 0.99, and we should be a bit worried because it's yellow. But you see a box that says optimize. And if you click that, then we do a short refinement for you of the ligand protein complex. And then when we have finished that, you get the view on the right-hand side, where you can download this optimized protein ligand complex as a structure file. And you also immediately see that the class score has substantially improved from 0.99 to 0.29. Another feature that we provide is that you can now fill your own models. So when you go to the AlphaFill web interface, you can upload your own model. Please make sure that it is a valid MMSIF or a PDB file. If you want to run a bunch of structures, you can also download AlphaFill from GitHub, install it locally and run it there. Or you can use the AlphaFill API, which will be there soon. So let's go to some structural examples. So here we're looking at the ABL1 kinase with an ADP bounce. This is the AlphaFill model, and we see a local RMSD of 0.99, which is a bit worrisome, not that bad, and the clash score is fine. However, to the same protein, we also transplant an ATP molecule. And we see that even the scores improve a bit. So we see that the local RMSD goes down and also the TCS goes down a little bit. And this indicates that the ATP bound state might be a bit more suited than the ADP bound states, indicating that AlphaFold has predicted a state of the protein that is closer to the inactive state of this kinase than the active state of the kinase. When we look at the zinc binding site in this stem binding protein, on the left-hand side, we see that there is a properly predicted zinc that is involved in protein function. It also originates from a very high sequence identity, and it had good scores, so it's not really a surprise. And the same holds for the zinc on the right-hand side, which is involved in structure integrity. You see a typical zinc finger there. When we look at those two zinc atoms transplanted to the zinc finger protein 91, we see on the left that there is a zinc atom properly transplanted, but it originates from 36 sequence identity. Still good, scores are good. The zinc finger itself looks fine, nothing to worry about. But the zinc on the right-hand side does look a bit odd, which is also reflected in the local RMSD. And this is illustrating one of the limitations of the AlphaFill algorithm, that it does rely on homology, because it originates from 35% sequence identity. So yeah, not much you can do about it. It can also happen that there are no close homologs available for the AlphaFold model. And then you will end up with an empty AlphaFill model, or in other words, we cannot transplant anything because there are no available homologs. And that made us wonder if we can use artificial intelligence to recognize ligand binding pockets from structural patterns. We do have sufficient data. There are over 100,000 ligands binding pockets in PDB redo and over 12 million in AlphaFill. So data-wise, it should be fine. So we thought, well, let's start with simple cases, metal ions. We chose them because they are important in structure stability and enzymatic reactions. And they are single atoms, so there's no rotation and also only one conformation. So we went on and we selected all the most commonly occurring metal ions, namely zinc, magnesium, calcium, iron, and copper. And we selected a training set and a validation set for all those metal ions. So for zinc, magnesium, and calcium, we have 4,000 for training and 1,000 for validation. And for iron and copper, we have over 300 for training and about 30 to 40 for validation. But a neural network does not really know about protein, atoms, etc. So we need to convert the 3D coordinate system into a grid space. And we do that by first defining 9 chemical properties of atom types. So we group all carbons into one category. We have 3 types of nitrogen, so the nitrogen and histidine, the positively charged nitrogen atoms, and other nitrogen atoms. For oxygens, we also have 3 categories, the hydroxylic oxygen, the carboxylic oxygen, and the carbonyl oxygen. And for sulfur, there are 2 sulfur types usually in proteins, so we define the sulfur and methionine, and the sulfur and cysteine being a different type of sulfur. Then, with these atom types assigned, we transform the atom position from Cartesian space to grid space. And to illustrate that, please look at the grid below. So the carbon's coordinates are put into the grids, and then we also select the surrounding grids. And we do this with a grid spacing of 0.5 angstrom. And the same for the oxygens and all the other molecules that are in the protein, all the other atoms that are in the protein. But then the neural network needs to learn how a metal binding site looks. In order to do so, we place the metal ion in the center of a container cube with size of 36 angstrom. And then we randomly take a sampling cube, which has size of 24 angstrom, as you can see here. And then within this sampling cube, we define a learning cube, and this learning cube has size of 15 angstrom, and it's always in the center of the sampling cube. Then we can start the learning process, which we do over 100 epochs. And first of all, we randomly rotate the sampling cube, which is then passed into the neural network, and we use UNet++. So it takes the sampling cube as input, and it goes through some convolution, and it returns a probability grid per metal. So we get 1 for copper, 1 for zinc, iron, calcium, magnesium, and also 1 for an empty grid. We performed learning on over 10,000 proteins with in total over 38 metal sites. So for zinc, we have almost 14,000, calcium 8,500, magnesium 13,000, and for copper and iron, almost 1,500. What we then do is that we search a protein for metals, and then we define the sampling cubes and the learning cubes. We feed it into the network, and then we get the metal probability back, predicted in those sampling cubes, as shown on the right-hand side. And then we reassemble the learning cubes into the prediction grids. Next, the grids are filtered, and the atom positions for metals are defined by the selection criteria. So when we have performed the filtering, we get this kind of gray blob that you see in the figure when the criteria are met. And the selection criteria are that there should be at least 2 amino acids per probability area, and the thresholds for probability are 0.99 for calcium, copper, and zinc, 0.97 for magnesium, and 0.8 for iron. You can see in this example case that for this binding site, we predict the zinc, which chemically looks okay. The neural network that we trained has currently an overall accuracy of 0.91 and a precision of 0.75. So let's go to some examples. Here I show some correctly predicted metal ions for this DNA-related proteins. So on the left-hand side, we see a correctly predicted zinc. In the middle, a correctly predicted iron, and on the right-hand side, it's a calcium. And what's even more striking is that the neural network is also able to define bimetal binding sites. So in this case, we find a bimetal copper site for it in hemocyanin. There are also things that go not that good. So here I show an example of a false positive where there is magnesium predicted by the neural network, but there is no metal ion in the PDB redo model. It does look like a metal site though, so we cannot say that the neural network is wrong, but it's not there, so it is a false positive. Then here is a false negative, where in the PDB redo model there's a copper, but the neural network does not predict any iron. And then there's also a third category of things that we notice is that the neural network predicts in this case a zinc binding site, whereas in the PDB redo model there's a nickel. Obviously, the neural network has not learned about nickel, so it would never be able to predict it. But there are some false identity cases within the atom types that we use as well. Ultimately, we want to implement this algorithm to AlphaFill, so we should also be able to predict metal binding sites in AlphaFold models. And this week we have tried some. So these are really the first results. But we do see that within the AlphaFold models, the algorithm is also able to predict, at least for those two proteins, proper zinc binding sites. At least they look like zinc fingers. The side chains might need a bit more optimization, but that is something due to the static use of the models. So with that, I would like to summarize and with the take-home messages. So I think we can all agree that the AlphaFold database has a transforming effect on structural biology research. And with AlphaFill, we created a new resource to enhance biological interpretation of the AlphaFold models. The AlphaFold models are enriched with ligands based on homology, and we can also predict metal binding sites and protein structures. This prediction algorithm needs some more validation and testing, and then we will implement it in AlphaFill. And afterwards, we will try to see if we can predict binding of larger molecules into the AlphaFold models and other proteins. And that leads me to the acknowledgement. First of all, I would like to thank Maarten Hekkelman, who wrote the AlphaFill algorithm and built the database. Velankar is working on the metal binding prediction, and Robbie Joosten and Tassos Perrakis are guiding and supervising this project. The funding comes from INEX Discovery, EOC Life, and OnCode Institute. And if you're curious, please have a look at your favorite protein. You can not only look at the proteins that are in AlphaFold, but now also upload your own models. They are not in AlphaFold yet. Thank you for your attention.
First questions.
Sally Chambers, and I'm Digital Humanities Research Coordinator at Ghent University, and I also work at KBR, the Royal Library of Belgium, on digital cultural heritage material. Inspired by our colleagues at the Royal Library of Denmark, we're really keen to see how cultural heritage material can be computed using the supercomputer. For example, we have hundreds of years of digitized historical newspapers, but they're not necessarily machine readable or have been split into articles. The supercomputer can help, together with computer scientists, to train models in order to extract the knowledge that humanities researchers need. The VSC has been amazing in helping Claria, our humanities research infrastructure, in order to provide both access to the data, so for example, the digitized and born digital resources from the Royal Library of Belgium, and with facilities such as the Jupyter Notebooks, can also help humanities researchers who are less computationally savvy to be able to still work with the digital cultural heritage data and do initial analysis and gain more confidence in being able to use supercomputing services. My dream is that we could have access to all European digital cultural heritage content on a supercomputer like the VSC, so that humanities researchers can analyze it. The VSC is helping us make that first step a reality.
Hello, I'm Álvaro Cortés and I'm bioinformatician at the Genomics Core Leuven. This is a shared facility between the University Hospital and the KU Leuven that facilitate the access of sequencing technology as well as supporting the data analysis and the data processing of DNA sequencing data. The purpose of the Genomics Core is twofold. We provide support for molecular diagnostic at the UZ Leuven and other hospitals in Flanders, and we support the sequencing workflows of research groups within the KU Leuven and other external groups. Before the VSC, we had to rely for both diagnostics and research on the internal cluster of the hospital. This posed unnecessary stress on our internal systems, as different projects were competing for limited computational resources. We then had to be very careful on how and when to schedule high-demanding computational jobs, as for instance de novo assemblies or whole genome analysis. Additionally, an increasing number of researchers wanted to analyze their data themselves, and they didn't have enough computational capacity to do it. With the VSC, we were able to move some of our complex research projects to the cluster at the VSC, alleviating the internal load of our own systems. We have successfully performed de novo assemblies, long-read iso-seq analysis, and RNA-seq analysis. We have also guided our research users on the use of the VSC to perform complex genomics analysis, which enable them to advance their science. The analysis of sequencing data is a demanding computational task. The VSC offers all computational capabilities to analyze such a data efficiently and with excellent assistance from the support team. If your research is in need of such analysis, the VSC would be my first recommendation.
My name is Wim and I'm a climate scientist working at the Vrije Universiteit Brussel. In our team at VUB, we use global climate models to study how climate extremes will evolve towards the future, and also to better understand the role that the land plays in shaping these climate extremes. So in our group at VUB, we use the Community Earth System model, which is a global climate model that represents all these climatic processes that occur in the atmosphere, the oceans, the land, and the ice sheets. Now, as a group, we contribute to improving the quality of that climate model, by introducing and improving the representation of water management and land management in that climate model. For instance, we incorporate reservoir management or irrigation techniques in that climate model. And by doing so, we improve the quality of the model, we make it more realistic, and this helps to generate more reliable future projections of climate change. So to run our climate model, we need access to high-performance computing infrastructure. And when we have that access, we can contribute climate simulations to the international climate science community. We can thereby support the development of the IPCC climate reports and also contribute to efforts such as the one shown behind me, where we quantify how many climate extremes a person born in a particular year in a particular country will face integrated across their lifetime. So doing this type of supercomputing simulations allows us to make international contributions to climate science. VSC is critical for performing our work as global climate modelers. With the help of VSC, we can run our global climate model on the supercomputer, and we can store the output on a shared data storage facility for further analysis. We are also collaborating with a whole range of other research groups in Flanders to share these resources for climate science analysis. And by doing so, we can improve our understanding of climate change, and we can help contributing towards solutions.
Today we are here at the Faculty of Veterinary Medicine of Ghent University. My name is Sebastiaan Teuns. I'm a veterinarian and co-founder of PathosSense, which is a new spin-off of Ghent University. With PathosSense, we offer complete diagnostics of infectious diseases for veterinary medicine. In this, we use nanopore sequencing, a revolutionizing way to analyze DNA and RNA of different kinds of organisms, including viruses and bacteria. Now today, Nick Verreke, my colleague, will take you along in his research, in which he is combining nanopore sequencing and a lot of different computational analysis for which he uses the Flemish supercomputer.
We are working with nanopore sequencing. We started with this small MinION device, and recently we moved to the GridION, which allows us to generate a lot of more data. This sequencing data can be used for bacterial identification and antimicrobial resistance testing, which later on can be used for quick and real-time bacterial diagnostics in veterinary medicine. Could we have done this without the use of the HPC? Probably yes. While we started with a CPU-driven computer, we quickly went to a GPU-driven one. However, this did not sufficiently speed up our processes. So we went to the HPC, which could actually facilitate our programs and algorithms to speed up our analysis even further. VSC allows us to focus on the science and less on the computations, so we can improve our portfolio and the number of analyses within the spin-off company.
Thanks to the Flemish Supercomputer Center, we have been able to bring our research to the next level.